He's also authored 2 books. For 20 years, the Ecological Integration Symposium has been about uniting many different fields in ecology right here on the A&M campus. Throughout his career, Dr. Marra has done just this, and today we have the honor of hearing about some of his work. Everyone, please welcome up Dr. Peter Marra.
Thank you very much, Meredith, and thank you everyone for inviting me here today and listening to what I have to say. Make sure this gets going. So in a lot of my work, what I've done is basically use birds to ask questions, use birds as sort of a model system to ask questions about how humans are impacting the earth. And I bring into my work a lot of different tools from biogeochemistry to engineering to physiology to fundamental ecology. That's not the first slide. You're showing, giving me the answer. But a lot of what I do is really using fundamental ecological or fundamental science approaches to address conservation issues. I'm really interested and passionate about just doing fundamental biology, but I don't think we can do really impactful conservation unless we bring in really fundamental ecological theory. And so I try to combine the two as much as possible throughout what I do. One of the things I want to emphasize is that all the work that I do is highly collaborative. None of the work I do is by myself. And I've had just incredible, patient and brilliant mentors throughout my career, which I feel really thankful for. Enormous numbers of colleagues in a variety of different disciplines that I work in. I can't become a biogeochemist, so I work with lots of biogeochemists like we all do in how we collaborate. So, and incredible colleagues and postdocs and graduate students and field technicians and students of all types from all over the world to do the work we do. And I don't even list them because I don't want to forget anyone. But there's just so many folks that have contributed to the work that I'm talking about today, or even the work that I've just done throughout my career that I'm not going to be talking about. This is certainly true in a paper we just published 2 years ago on the decline of the North American avifauna. And this is basically the process of the 6th mass extinction. That's what this is. Species decline before they go extinct. And we are documenting this decline in many different ways. And what this group of colleagues and I documented was basically a net population loss of 2.9 billion birds using a variety of different monitoring tools. We're lucky with birds in that we have so many different sources of information from, the community contributions through citizen science to long-term monitoring through things like the Breeding Bird Survey and Christmas bird count data. And then there are federal counts of ducks. And we sort of brought all those things in to sort of assess and look at how populations of birds have declined over the last 40 or 50 years. This is a 29% loss of overall abundance of species. 303, or over half of the species in the US and Canada, have declined significantly. And this is where we've been able to do this. Other places in the world where people have started to look at this, unfortunately, we're finding the same thing happening. The problem is in most of those cases, we don't know why the species are declining. We don't know where or what or where we devote important dollars to implement action to stop the declines. So my whole career, one of the big threads, not the only thread, but one of the big threads in my research has been about what are the factors that limit and regulate population dynamics. This is a fundamental ecological question that gets at things as simple as survival and reproduction and mortality, density dependence. These are fundamental issues that they're of critical nature to how we do conservation, if we hope to do it effectively. And this is the big thread that's going to run through my talk today. And when you apply this question to migratory species, you have to bring in a bunch of different things to be able to ask that question and answer that question accurately and effectively. So today I'll talk about cycles. Cycles are something I'm really passionate about, and migratory connectivity. I'll explain what I mean in just a moment. Full annual cycle ecology and why that's fundamental, but it's only the first step. What we need to be doing is full life cycle biology, ultimately, if we can do it, of a species known as the American redstart, but I apply this question to many different species, but American redstart's sort of the one I've been working on for over 30 years now. And then I'll bring you down the road of looking at new technologies, new tracking tools, and how it's getting smaller and smaller, allowing us to ask more and more important questions about the biology and ultimately applying that information to the conservation of these animals. Cycles. So cycles are something that I've thought about for a long time. And I think when it really clicked that cycles were something that were important to me when I was taking an endocrinology class at LSU, not too far from here, with a guy by the name of Al Meyer, who was a graduate student at University of Washington with Farner and King, who are two of the guys. If you study endocrinology at all, Farner and King are two of these gods in the field that sort of came up with and discovered a lot of the important circadian controls and circadian cycles that are out there. But in the circadian world and in the biochemistry world, we've understood the importance of cycles for a long time. But cycles are pervasive in all physical and biological systems. They really are. And it's this fundamental driver of the tilted Earth spinning around an axis around the sun that sets billions and billions of things into cycles, from the biochemical to the cellular to the individual to the organismal level. Again, these are circadian cycles on 24-hour light-dark cycles. These are annual cycles, like the seasons, as we move from spring to summer to fall to winter. Animals and plants have to respond to these cycles of day length, of temperature, of a variety of things. These were important and the ultimate drivers of a lot of the biology that we see. And again, in the biochemical literature, in the endocrinology literature, people have respected this and understood this. In the ecological literature, conservation literature, evolutionary literature, not so much. And so I did a review of papers with several postdocs. We did a review of the papers in the various ecological journals for 4 major vertebrate taxa, amphibians and reptiles, birds and mammals, and looked at when these studies were conducted, what season, how many of these studies just looked at multiple seasons, and how many of these studies looked at something we've been calling a seasonal interaction. That is, your current state, to what degree is your current state influenced by previous states? It's that simple, and it's that logical, because you are who you are today because of where you've been and some genetics, no question about it, but we're influenced by our environment. So when you do that, what we found is that the majority of studies were done during the breeding season, ignoring things in transit and the stationary non-breeding season. The vast majority of studies also focused on one period of the annual cycle, and almost none, or very few, when this was done in 2013, I guess, looked at seasonal interactions. These numbers are really increasing. In fact, the number of papers looking at seasonal interactions is increasing dramatically. So there's clearly a bias in our research and in our understanding of the fundamental biology of vertebrate taxa, and I think if we expanded this out, in many taxa, in how we understand the events throughout the annual cycle or throughout the life cycle of an animal that influences its fundamental biology. So why is there a bias? Well, I think there's multiple reasons why there's a bias. It's logistically challenging. It's a pain in the ass, folks. It really is. I do it. I've done it my entire career. It's not easy. It's expensive, and there's also a timing thing. Most of the research is done from academic institutions. And when you work within the academy, when do you do most of your research? Done during this, in temperate areas, which is, again, where most of the research had been done historically, it's in your summers. And that's when things are breeding. And so I think there's been this sort of unintentional bias that might have introduced when things were happening. It was very hard for me as a graduate student to go and do work where I did my work in Jamaica during January through May or in the fall, because I had to TA or I had to teach or take classes. This makes understanding the biology and the inherent biases that are introduced quite complex. But there is also a false impression. There has been a false impression that things outside the breeding area is unimportant. In fact, I've heard really famous ecologists say this, that anything outside reproduction doesn't matter at all. But that's out there. That was out there. I think it's changing, but that was out there. But there's something else, especially with migratory animals, but anything that perhaps moves, perhaps not Richard's plants, but except when they go underground, but certainly with things that move long distances, there is this difficult thing about following them. So we have a poor understanding of something we call migratory connectivity, in part because we haven't been able to follow these individuals because they're too small, they cover much larger areas, and so we can't track them. It's that simple. It becomes difficult. So we can't follow these individuals. So we're forced to sort of capture them in this one short period of time, and then we sort of try to figure out all these other things, ask all these other questions from this little glimpse of period of time that we get to handle the bird. or the animal, whatever it happens to be. We let it go, we see it again, we take a sample to try to understand its tissues and where its tissues tell us it's been or whatever it happens to be. But if you think about the amount of time we actually know where the animal is and then the amount of information we try to derive from that, it's kind of interesting. So this idea of migratory connectivity is the geographic and temporal linking of individuals or populations between different periods of the annual cycle, including migration, breeding, and non-breeding. Migration being a non-breeding period, but oftentimes there's a stationary period of that. But linking in that and then understanding where this animal goes, it's exposed to a variety of things, whether it's competition, whether it's an environmental thing, it doesn't matter. But doing that throughout an annual cycle and ultimately throughout a life cycle is really where we need to be. But we're really behind the 8 ball with just understanding migratory connectivity, I would say, for most animals. So knowing this is critical. And I want to describe to you a few reasons why it's so critical. First, I'll talk about just fundamental movement ecology, which is sort of a new paradigm, which I'm kind of torn about because I think it's just really ecology, frankly. But it does define an important understanding of what happens to animals when they move. Let's see this. Great. So this is Autumn Lynn Harrison. She runs a project for me called the Migratory Connectivity Project. She's just captured a black-bellied plover on the north slope of Alaska. It's a beautiful shorebird. You see the 3 eggs. We're attaching a 5-gram satellite transmitter to the back of this bird and letting it go. Now we get to observe the bird a lot. So we're on the Colville River Delta. Bird just finished breeding. This is just one bird. There's thousands and thousands. Bird's fattening up. Getting ready for the first big one. Staging area. And it's fattening all the way down, it's up again, across the Gulf of Mexico, into Central America. Wintering grounds. Grew for several months. Avoiding peregrines. There's so much we can learn remotely about how birds migrate. Rhode Island, stages again. Right back up to its exact again. Breeding. Same species. Gulf of Baja. All the way to Peru. Caribbean. The exposures there are incredible. If you think about the variation in environmental exposure that these animals are experiencing, or even environmental threats from humans, we want to understand their population dynamics, understand the mortality, understand what things influence their reproduction. It's quite complex. So that's an example of movement ecology. We have more and more. We've all seen these incredible sorts of visualizations that put into context, into perspective, the scale at which these animals move and the scale at which we need to be thinking about from a conservation perspective. So let's look at something a little bit more real from an applied perspective. This is the yellow-billed cuckoo. This is a project with a postdoc of mine by the name of Callie Stanley. Yellow-billed cuckoos are a species that have been declining precipitously throughout their range. There's a western species that was just listed, western subspecies that was just listed. You don't even see it on the map anymore. There's just a couple sites where we were able to capture them, but those are considered the westerns. So we've been putting satellite transmitters. These are 2-gram satellite transmitters. The one, the black-bellied plover, was a 5-gram satellite transmitter, but now we were actually using 2-gram satellite transmitters. These cost about $5,000 a piece. They cost $1,000 a year roughly for transmission charges to the Argos satellite. They give you about, just so you know, about a 1 kilometer resolution in data. Sometimes it's a little bit better than that. but we've been putting them on these birds for a while. We know where these birds breed. We know that they're bleeding across the landscapes in terms of their population trends. They're just crashing. But we didn't really know where they winter. They have a very complex species relationship with resident species throughout South America. And what we thought was going on and what we found were quite surprising. So these are some of the tracks that we've gotten from yellow-billed cuckoos. You can see these eastern populations really bolted down to an area known as the Gran Chaco. This was southern Bolivia, northern Paraguay, and northern Argentina. And it was funny, at dinner last night, I was talking to Lee, and Lee's like the granddad of the Gran Chaco. I had no idea how long he had been working there. It was really spectacular to get his insight into this area. But we've been starting to work there remotely using satellite tags, not having gotten down there because of COVID. We've just finished this the satellite tracking data, and we're now going to go there. So this was the previous range, purported range of the yellow-billed cuckoo. And what we've discovered is that's actually the range of the yellow-billed cuckoo. It's really quite remarkable. And it's kind of funny because most of you are probably familiar with eBird from the Cornell Lab of Ornithology. It's a remarkable citizen science project. So we went in and looked at the eBird records and modeled the distribution of the yellow-billed cuckoo using citizen science data. And that's what we got, which is pretty remarkable too. It's like, oh Jesus, I spent a lot of money on tags and just to confirm what the eBird's already told us. So anyway, don't tell anyone that. And what else is really interesting here is that when you look at the Westerns versus the Easterns, it turns out that the Westerns are in the southern part of the Gran Chaco and the Easterns are in the northern part of the Gran Chaco. So there's actually this subtle segregation that's going on between these two subspecies. And it's not that surprising if you think about it. I mean, the Westerns are actually using a totally different migration route. Temporally, they're totally different than the Easterns in terms of when they arrive here, how they migrate. So the fact that they're segregating there, it kind of makes sense. And so now we're going to go in and capture them and look at genetics and a whole bunch of different things next. But when you're going back and asking questions about conservation and looking for the smoking gun, the easy smoking guns here, which is it's tricky. It's really hard to understand what specifically is limiting a population. If you haven't thought about it, I encourage you students to think about this. It's a really tricky thing. But when you start to look at the remote sensing of these areas, what you find is that this area has been decimated for soybean and cattle ranching. It's been just destroyed, especially the area that represents the area that we think is primarily used by Western cuckoos. So these are areas that the cuckoos stay in for 3 to 5 months of the year. It's a long time. And if you're decimating the habitat, guess what? Cuckoos go poof. And that's what happens. It influences their condition. It influences their survival probability, their ability to survive in a migration. We don't know yet where the mortality happens, but there's probably some interaction that's going on there. And this has been reported in various places, but it just hasn't got the attention that it needs to get. And so we're going to continue to work on cuckoos down in the Gran Chaco. But this is a really important example of where understanding fundamental movement ecology tells you something about the conservation or the vulnerability of a particular species. So we talked about movement ecology, conservation, and now I'm going to talk more broadly about population biology, population dynamics of a species that I've spent a long time studying, the American redstart. And this is the basic life cycle or annual cycle of an American redstart. They have a short migration, which is starting right about now in April and May. They're leaving the wintering grounds in mid-April in places like the Caribbean. And they go on a pretty quick migration, 2-week migration, depending upon where they breed. And they breed from Louisiana all the way up to New Brunswick and Nova Scotia, all the way west to Montana and Washington and southern British Columbia. But they have a pretty quick breeding season. It's 2 to 3 months. They're single obligate nesters, only one. They don't do any double brooding. It's never been documented. And then they go into a molt. So males arrive first, females come, choose mates. There's polyterritoriality, polygyny in the species, but no double brooding. They go through a pretty quick molt and they bolt back down to the wintering grounds where they spend the majority of the annual cycle. And so what I'll be talking about today is how these events, what happens during the breeding season, is not just a factor of things on the breeding grounds, it's influenced by things happening thousands of kilometers away, which as you think about it, makes a hell of a lot of sense, right? So I'll just basically show you that's what's going on here. And what I'm talking about are these things called seasonal interactions. There are circumstances within one period or season of the annual cycle that influence events or traits in a subsequent period. And how I've separated these are into ecological or evolutionary seasonal interactions. Whereas the ecological are basically at the individual level, where you have things known as carryover effects, which are non-fatal things like that influence the timing. It might be poor food availability, dry winter, that doesn't kill an animal, but it causes some sort of an impact on a subsequent period. It might delay migration, delay its arrival in the breeding grounds, which is basically what I'll be showing you. And it influences the number of young that individual will fledge. Or it might influence some other factor of its molt that influences its ability to fly. There are an infinite number of ways these things can manifest themselves. This also happens at the population level, where you might have really high reproductive success during one period of the annual cycle, and you have negative density dependence that might be happening at a subsequent period of the annual cycle, or really high overwinter success or survival, and then you have really high density back on the breeding grounds, and so you have negative density dependence on reproductive success on the breeding grounds. These are carryover effects. These are seasonal interactions at the population level. And this happens in evolutionary terms as well, where you might have a trait, a morphological trait, wing length, color, that are selected for throughout the annual cycle through a series of trade-offs over time, over multiple generations. And this is how we have to be thinking about these things, even sexually selected traits. Just had a student finish 2 weeks ago at University of Colorado Boulder working on barn swallows with Becca Safran. And basically she was showing that these traits that we see, these colors that we see on these birds are things that are evolved over multiple generations, but it's selection that's acting not just on the choice of a mate, but also its ability to survive in the non-breeding grounds. So this is the model basically that we've worked from in our project down in Jamaica, which this is our 33rd year. I've got a student there, right, postdoc, and several students there right now. Oops, didn't come out quite right. But basically the idea is that there are a combination of density independent and density dependent factors which drive the events during the non-breeding season that influence seasonal interactions and carryover effects between our breeding population and our non-breeding population. So habitat is influenced by rainfall, and rainfall basically affects food and habitat directly and indirectly. This can influence bird density directly. Sorry about this. This was a transition issue. Or food can influence body condition directly or through density and crowding. And that will eventually influence either overwinter survival or survival during migration back to the breeding area. So we'll come back to this a few times today. Redstarts have been a really wonderful species to study over these years, in part because they consume entirely insects. They're pretty much 100% insectivorous. Males and females hold territories for the most part. We're starting to see there's a lot more play here than we thought before. And they're easily aged in sex, which is important for demographic studies. So you can actually catch them and watch them pretty easily. The work is in Jamaica, although I studied them throughout their winter range. Again, they've got a fairly wide breeding range from Louisiana, you can see how wide it is, and a fairly large wintering range throughout the greater Antilles, a little bit lesser Antilles, Pacific coast of Mexico, down through Central America, Northern South America. The study sites that I focused on are in this area of Jamaica. It's a much less developed part of Jamaica. We work in primarily 2 habitat types there, although redstarts occupy multiple habitat types, but we're focusing in on 2 habitat types just for our experimental design. So a black mangrove swamp like this, that again, as the birds arrive early in the fall, it's really moist, it's really wet. This was a wet year, so when I went down there in February, we were up to our waist wandering around in these mangrove swamps, which also have quite a lovely crocodile population. But as the dry season progresses, it does dry out, as you just saw. And it's neighbored by a series of scrub type habitats that dominated by a logwood tree. It's a non-native tree. But as this drying happens through the typical tropical seasonal effects, these trees drop most of their leaves, and this area becomes quite harsh, unlike the mangrove, which stays fairly lush and moist. And so we study redstarts, males and females, young and old, in both habitat types to control for age and sex, and really to understand the effects of habitat. We do a variety of things. So we capture them, we weigh them, we pull feathers from them, we pull blood samples from them, we dance with them, we do everything with these damn little birds. And we weigh them to the nearest 1/10 of a gram, and we color band them too. So they all have their own unique color bands on them. Now we actually put little radio tags on them, MODIS tags. So we're actually automated a lot of this work now, believe it or not. And we recapture them multiple times to weigh them again, to look at how their mass changes. If we're bleeding them for hormones, we'll reassess hormone levels like corticosterone and triglycerides, other metabolites of various things, depending upon the question. We recite them and recapture them within seasons, as well as across years, to be able to estimate apparent survival of these birds. And then we measure food through a variety of techniques, mainly arthropod sweep netting, to really assess how the food availability might be impacting various traits here. Was going well. There it goes. Not there yet. So I'm just going to tell you the answer. Performance does depend on habitat occupancy, regardless of your age or your sex. So if you're in black mangrove or a black mangrove habitat, food availability is usually high and stable over that entire winter period. In scrub, you get a serious, significant decline, not surprisingly. We've measured this in so many different ways. There's just massive decline. And not surprisingly, body condition changes as well. Just mass change, and these birds have no fat, very little fat. They're lean, mean muscle machines. They really are. And so if you're in black mangrove, regardless if you're male or female, young or old, you maintain your body weight in most years. Not in all years, but in most years. Whereas if you're in second growth scrub, your body mass is changing, again, with males and females, et cetera. Your departure date, no one's really measured this before, but we've actually been measuring it for 33 years now. We quantify the departure on spring migration for our entire population. You leave significantly earlier if you're in black mangrove, regardless of your age or your sex. And in scrub, your departure is delayed. And same with survival. Survival's high, regardless of sex and age in mangrove, and it's low in the second growth scrub. And we've done a variety of experiments in this system as well. So we've done, Colin Studds, a former student postdoc, and I did a removal experiment where we removed mangrove males and replaced them with females from the scrub. And we knew that they were coming from the scrub using a variety of techniques, both color bands and stable carbon isotopes, where I'll come back from, which is a signature of habitat. I'll describe that in just a moment. But when we upgraded these birds from the scrub habitat to the mangrove habitat, those upgrades relative to controls maintained their body mass, left significantly earlier, and had higher return rates between years. So there's clearly a habitat effect there. We've done insecticide, just insect removals or depletion experiments where we've basically shown the same exact things. And we've actually done irrigation experiments. Turns out when you water plants, they keep their leaves. It's pretty much what we show. This was one of the most challenging experiments I've ever done in my life. It was with Scott Wilson. That was Nate Cooper with the insecticide work. And it was really, really challenging. And we did get a leaf effect and we got an insect effect, but no bird effect, just because the area that you have to irrigate is massive. And this is just a tremendous pain in the ass. If you ever want to do this, call me. And I'll save you millions of dollars and all kinds of stuff. I mean, it's fun, but it's a lot of work. So I've just focused on habitat there, but since this is a long-term study, we're also able to look at the effects of dry years versus wet years, because this area is subject to El Nino, La Nina effects. And so there are extremes. And not surprisingly, year does substitute for habitat. And so March rainfall is on the x-axis, arthropod or biomass, food biomass is on the y-axis, scrub is in red, mangrove is in blue. So mangrove is always has higher biomass than scrub, but as you get more and more rain in March, as an indicator of the spring, you basically get more and more insects. Add water, get insects. That's pretty much it. And if you look at corrected body mass, which is an index of body condition in general against March rainfall, this is sort of a bird that would be in the optimum, basically the average condition across the entire winter. So if you're below the line, you're in poor condition. If you're above the line, you're in good condition. In general, birds in mangrove are in good condition. Birds in scrub are in poor condition. But you can see it goes up as it gets wetter and wetter and wetter, depending upon those years. And this influences departure date, not surprisingly. So body condition on the x-axis, spring departure here. Birds in scrub tend to be leaving later than birds in mangrove, and this is influenced by annual rainfall. So it's clear that there are a whole suite of density independent factors that influence body condition and departure. I haven't even gotten into seasonal interactions yet, but we'll be getting into that in just a second. This is sort of the lead up. You've got to understand what's going on in this state to understand the subsequent state. So let's ask about density dependence. No one really likes to study density dependence. People don't study density dependence. It's a hard thing to study, but it is fundamental to so much of what we need to understand. It really is. And there's been very few studies during the non-breeding season for any organism that I'm aware of. There've been a few, but very few. So we asked the simple question, does red start survival decline with density? And it turns out it does. This is for ASY males. Older ASY is after second year or older. We can age them as first year or second year, but then some of these birds we track for 11 years. We have individual ages for many of these birds. And this is females. And in both, you can see that there is, as density increases, apparent annual survival also goes down, which was quite surprising, in part because overwinter survival is constant. So the mortality isn't actually happening during the winter period. It's happening at some subsequent period after this, and I'll come back to that in just a moment. In scrub, it's a weak negative density dependent feedback, not significant, but it's quite weak, but there's still an effect there. So we asked that question, what's really going on? Does red start condition decline over winter since we're not finding an overwinter mortality effect? Does that make sense? We're not seeing mortality then. So there's got to be something going on there. So we asked that more subtle question that we've been doing, is condition declining? And sure enough, when we did this with birds in mangrove, again, the transition didn't quite work to PC, but this is density here. This is that corrected body mass as an indicator of condition. Birds in mangrove, their condition declined with density, as we predicted. But in scrub, it's like, what the hell? Something totally opposite happened, which was really actually quite interesting. We didn't predict this at all. As density increased, mass got better and better and better. And what we think is going on here is that birds are actually limited by density. There's real density dependent regulation going on here. But here, birds are settling to meet the carrying capacity, the increasing and improving carrying capacity of the habitat, but they continue to be limited by food, not density, which was a quite interesting nuance to this that we weren't expecting. So it's clear that there are density independent, which shouldn't be that surprising, and density dependent, some combination of these two that interact to ultimately structure these processes during the non-breeding season. So what about things post non-breeding? We now have a good understanding of what's going on here. How do we make that jump to understand where these birds are going? And frankly, we didn't even really know where these birds were going at this time. We thought they were kind of going, holding our breath and hoping they were going up towards our breeding area, which was, I was working on them at the Queen's University Biological Field Station and up at the Hubbard Brook Experimental Forest. We kind of thought it was that latitude, but we weren't totally sure. But we asked the question, does non-breeding habitat occupancy or precipitation influence reproductive success? And this is something I did way back for my dissertation at Dartmouth. And at the time, I was really interested in trying to figure out all these different measures of condition and how we can sort of assess and sample a bird, take a snapshot, figure out how it was doing, how it's physically was doing. And there was a paper that had been published by this guy, Keith Hobson, on nitrogen, and was showing that there was nitrogen deposition when you had muscle catabolism going on. And I thought it was neat. So I thought maybe I could just sort of take a blood sample, look at nitrogen, and figure out what was happening. And when I did that, when you run this in the mass spec, you get nitrogen and you also get carbon. And I looked at the nitrogen data and it was like, shit, there's nothing there at all for nitrogen. But I looked at the carbon and the carbon was just striking. It was so different between the scrub habitat and the mangrove habitat. And I started scratching my head, and turns out that across a moisture gradient where you have variation in C4 versus C3 plants, or even if you have the same plant in different habitats that vary in moisture, because of the amount that those plants keep their stomates open, they retain some of the larger molecules of carbon or release some of those larger molecules of carbon. That influences the stable carbon isotopes that are retained in those tissues. Guess what eats those tissues? Unless they've got a lot of toxins. But the insects eat those tissues. And what do redstarts do? They eat insects, and they incorporate those tissues, those stable carbon isotopes, into their tissues. And that's basically what I accidentally fell upon, was that there was this pattern of stable carbon isotopes across these two habitats that acted as a wonderful signature that persists for 4 to 6 weeks. And so it was entirely serendipity, nothing there in terms of brilliance. It completely fell into my lap that there was this really remarkable pattern across these two habitats. And I sampled these birds in multiple places, not just in the wet lowland forest and black mangrove and second growth forest of Jamaica. I was so excited about this pattern, I wanted to go somewhere else. So I convinced my advisor, Dick Holmes, to give me some money so I can go to Honduras, anything to travel. And I sampled redstarts there too. And I was able to show that this pattern was actually quite persistent. And since this happened, we've looked at this all over the place. Lots of people have. And it is a pattern that is expected. But what's neat is that you can actually then catch these birds, not the same birds, not knowing where these, or even know where these birds are coming from. And you can sort of assess on the breeding grounds, what type of habitat did they come from? Did they come from a dry habitat? Did they come from a wet habitat? So I sampled birds this is the initial graph that we looked at, up in New Hampshire at the Hubbard Brook Experimental Forest. And sure enough, early birds versus late birds. These are birds coming from wetter habitats, birds coming from drier habitats. In general, there was a tendency for birds coming in later and later and later, which would also be birds leaving later and later and later, and generally probably from the same vicinity because they show high connectivity, coming from drier and drier habitats. So that pattern held up remarkably. Again, we've since done this at many, many sites with many, many species, and we're finding very similar sorts of patterns. And we've documented this with other things too. But you can then follow the reproductive success of these birds, put it into a structured equations model. And basically what we've been able to show is that in general, in most years, the total number of offspring fledged is most explained by the habitat you originated in during the non-breeding season, by 1 to 2 young, which is pretty significant, especially for a single brooded species. Again, we've shown this for Kirtland's warblers, we've shown this for ovenbirds, we've shown this for wood thrush, and people have shown this in shorebirds as well. This pattern is holding up. And again, now for ovenbirds, we're not tracking with isotopes, we're tracking with another tool called a geolocator to show that the same individuals, when they leave the non-breeding ground, determines when they arrive on the breeding grounds. And we're doing this with redstarts as well. But clearly, non-breeding season moisture and food determine departure and reproductive success. Again, we're seeing this over and over again. That is a seasonal interaction. But now we can start asking more subtle questions, more other questions about population level sorts of changes. And so the question that we're currently asking, and we haven't published this yet, this is fairly new, is does annual survival differ between wet and dry years from Jamaica, our study sites, and does this vary with migration distance? So after a dry year, does that have a differential effect on a bird from our Jamaica site that's breeding further south than one that's breeding further north? And does that differ depending upon it being a wet versus a dry year? Does that make sense? Now you're probably asking yourself, well, how do you know where your Jamaica birds breed? A different stable isotope. This time we're using deuterium. It's a stable isotope of hydrogen that varies latitudinally. And again, similar to carbon, it works its way up into the food web, and birds will actually ultimately molt this into their feathers where it is then inert. And they molt their feathers on the breeding grounds post-breeding. They fly down to Jamaica. It turns out I've been sampling these birds and pulling feathers for 33 years. So I've got a 33-year tail database, as does Tom Smith. And Tom and I talk about this. We've talked about it a lot at UCLA. But we can actually tell over time where and how our population has changed within Jamaica relative to where they breed, which is pretty cool. So what we found is after wet years, this is the modeled parent annual survival. This is the breeding location in the north, breeding location in the south, and this is the parent survival. This is after wet years. There's no difference in apparent survival after wet years in Jamaica. After dry years, it's a very different modeled apparent survival pattern. Northern birds have much lower survival than southern birds after dry years, suggesting that the range dynamics, like the northern edge of the range dynamics of the species, has less to do with the amount of habitat available in the breeding grounds than it does with what's going on in the non-breeding grounds. So range dynamics of this species is driven by things happening in the tropics. I'm guessing this is something that is much more common than we've ever imagined. And so when we do niche modeling and we look at predictions about what species are going to be doing on the breeding grounds, all that is primarily done with breeding season data, ignoring everything during the non-breeding season. And in fact, most niche modeling efforts are single season studies, only breeding season, and based upon temperature data, nothing else. So What's really interesting is that the Caribbean, like a few other places around the world, has been undergoing a severe drought, long-term drought over the last 30 years of my study. Every year, it varies. There's variability there. A lot of variability has increased. But in general, it's been getting drier and drier and drier. And so I have those 33 years of feathers. So we've just recently analyzed those 33 years of feathers and asked the question, has the mean breeding latitude of my Jamaican redstart population changed because of differential mortality on those northern migrants? And what we found was that's exactly what we found. That over these years from 1990 to 2015, these are young birds primarily. We didn't want to look at older birds because of dispersal. We looked at natal origin. In general, birds, as time has gone on, our population has moved further and further south. That basically, to put this on a map, this is we didn't really know exactly where our birds were coming from. And this relationship has gotten stronger. I'll show you in just a second why that is the case. But in general, because we're using isotopes of hydrogen, we only know the latitude. But this is what that difference represents in terms of how they've moved south. That's the difference there. But again, I just find this amazing that the northern range dynamics of these birds is driven by things in the wintering grounds, not by things on the breeding grounds. So to summarize this, I'm arguing that seasonal interactions drive fundamental biological processes, that we need to start thinking about all of our questions and reassessing, to what degree has this been influenced by things outside the period of time that I'm studying. I think you might be surprised at how, if you haven't done this, and many folks have, of course, how much of that variation might be explained by alternative events. It influences migration schedules, reproductive success, breeding season range limits. And I didn't tell you this, but we have 2 papers published showing that natal dispersal on the breeding grounds is driven by events during the non-breeding season as well. Natal dispersal is something that people think of as only as a breeding season event, but how far that individual moves from one breeding season to the next, people forgot about this 6 to 7 month period where they're actually in the tropics that influence things in terms of where they ultimately go. That's really important. Natal dispersal is a critical event. Ultimately, we need to be not only thinking about full annual cycle perspectives, but full life cycle perspectives as we automate and think more about how we can follow these animals throughout their full life cycles. But at least now we need more information on migratory connectivity. So we're actually at least studying the same populations throughout the year, if not the same individuals. Stable isotopes are fun. They've been really insightful and really important, and they continue to be. But tracking individuals, I got to tell you, is so much more satisfying. And so we're moving more and more into this with our studies. So I'm going to just show you a couple new things that we're doing. That's an American redstart in our site in Jamaica. And we started to put these light level geolocators on their backs, which are archival tags. These are tags that have a little light stalk on them. They have a clock inside of them, and they keep track of light exposure throughout the day. And they'll keep tracking for maybe 9 months. There's also a clock, so we know the time. Oops, sorry, don't want you to get seasick. And when you do that, you can actually calculate plus or minus 200 to 300 kilometers where that bird is, which is pretty powerful for what we can do with redstarts, because we never knew this before. And it turns out these little bastards have been spending all their time in like the Wisconsin, Iowa region, nowhere near where we had hoped they would be. And so they're in like the driftless region, which is really has a high density of redstarts actually. But now we're able to actually tighten up a lot of the relationships we're looking at. When we look at long-term monitoring data, we actually use those data now in a lot of our models, and it actually is tightening it up quite a bit now that we know exactly where our birds are going. We've also been working, another one of my studies, Kirtland's warblers, I work on with a colleague now, Nate Cooper, who was a former postdoc. Kirtland's warblers are this remarkable species that were down to 200 individuals in the 1970s and 80s. And they were placed on the endangered species list, and they started to remove cowbirds by the tens of thousands. And also they breed in jack pine, and jack pine is a fire dependent species. They started to put in a lot of prescribed burning, then a house burned down, then they started just to manipulate it mechanically, but they're managing the habitat intensively. And we're now back up to 4,000, 5,000 birds. We finally got them to stop the cowbird removal experiments. I can tell you, or removals, I can tell you about that. But what's so neat about this species is that we've been studying them on the wintering grounds, and there's a new technique out there for tracking birds. It's called MODIS tagging. It's basically automated radio telemetry, and it requires a terrestrial system of antennas. And all these white spots, white dots are antennas that we use in our tracking of this Kirtland's warbler population. But this area is just covered, littered in these MODIS towers. And if you have a small tag on a bird that are down, they're down to like 0.3 of a gram and has a long antenna, but it's a very, very light tag that lasts over a year now. As long as that bird flies within the range of the tower, it'll ping the tower. And these are coded tags, so they have unique codes on them. So I know the individual. But what's really neat is that we study them right here. So Kirtlands, they winter primarily on 4 islands in the Bahamas, the Cat Islands. They're starting to spread a bit, but those are the primary areas. We cover this area in Motus towers. We cover this area where they breed in Motus towers, and we pick them up here, but we then pick most of our birds back up here when they get to the breeding grounds with these towers, and then we go in with handheld receivers, and we actually catch those same birds again. So we're actually studying the same individuals now on the wintering grounds and the breeding grounds, and we've looked at things like microbiomes in the same exact birds, looked at how that changes. We can ask a whole suite of another questions, set of questions at a different level than we've never been able to do before, thanks to a new type of technology, which is it's been quite remarkable. This is the last animation I'll show you. One of the things that we're able to look at is how it happens over the breeding season with movement, sort of the secret lives of Kirtland's warblers. Each one of those is a bird coming back from the Bahamas off migration. Each one of these black circles is a patch of jack pine where they nest. And each one of these black things is a tower. They're just settling in now. They're breeding. They're pretty much paired up, a few late stragglers coming in, and they're incubating. So we're studying all these birds in the ground. We know exactly what's going on with the life history. And then all of a sudden, birds start to move. Turns out those are unmated males. And these are what we typically think of as floaters. But we've never really had the insight to understand how far these birds actually move. These birds are actually moving 20, 50, 100 kilometers, and they're actually doing it at night as if it's a migration. So the movement that's happening here was just really striking. There's so much more we want to know about this, but it was just so insightful by putting in this technology into this system to try to understand the spatial dynamics of a breeding season that we once thought was quite stable. Turns out that's not as stable as we thought. So that's all I had to say, and I'd gladly take some questions. Thank you very much. Yeah. Oh, sorry.
I'm going.
Hello, very nice talk. Thank you.
Sure. Thank you.
The section with the music in the background was very nice. Like your voice goes really well with the music.
I practice all the time.
Yeah. It's like a movie. So do you know like what determines choice, or I don't know if it's a choice, like what determines which birds end up at which wintering sites?
Yeah, in mangrove or scrub?
Yeah.
Yeah, it's a great question. Very poorly understood thing. And so I decided to do that for part of my dissertation. So I spent 2 years there from July until November, just quantifying the arrival dynamics of redstarts into these habitats. And basically what happens is it appears as though it's fairly random. So in the fall, remember that the 2 habitats are pretty much the same. The insect abundance is equal. Foliage it's wet, it's raining every day. It's a miserable place to do field work. But every day I went out there and counted birds. Turns out young birds typically come in first and settle randomly across the 2 habitats, and older birds start to come in, returning birds come in. And it's pretty much prior site dominance. And so the older birds come in and knock out the birds out of the mangrove. And the other talk I could have given you is on sexual habitat segregation, which is something I'm very interested in. These birds show really strong patterns of sexual habitat segregation, such that the birds in the mangrove habitat are about 70% male, 30% female. And in the scrub, it's about 30% male and 70% female. And some habitats are 100% male, some habitats are 100% female. And that's driven by behavioral dominance, primarily of males. But that doesn't happen immediately. It actually forms gradually over that arrival period as they start to battle things out, and older males come back and they start kicking out females and younger males, or poor quality older males.
Very cool. That's exactly the answer I was hoping for. Thank you.
You got it. Yeah, sure.
Great talk. I was wondering if on the breeding grounds, there's like mating between birds that had similar wintering habitats.
Yeah, that's a really good question. And I feel like we've looked at that, but I don't know if we have. It's a really good, I feel like we've talked about it. I don't know if we've ever sort of, looked at the data that way.
Okay.
Kind of want to think we have and we didn't see a pattern. That's my gut, but I just don't remember, to be honest. But it's a really important question. As we get into these, like with the Kirtland's warblers, as we, we're currently having big problems with working in the Bahamas. And so we don't have the study going on now, but I really hope that we can really dig in and start asking those sorts of questions with the same individual birds. Again, that's what I want to be, is same individuals, not playing with these isotope games or archival geolocators and have to recapture the friggin' birds. The same individual birds is the way to go. And if we can ask those sorts of questions with that, that's what we have to do. And using more of these tracking tools to do, again, same individuals. That's the way to do those sorts of questions.
Okay, I have a question because I'm controlling the mic. So no, I wondered, in terms of the species that we know there's sexual segregation happening on the wintering grounds, has that, the differential management of those different habitats, has that been reflected in populations of males versus females on the breeding grounds? Do we know?
Well, yeah, I think it does. And so in general, birds have a male bias sex ratio. I think it's primarily, because people have often said that it's primarily due to differential mortality of females on nests, because there's a 1-to-1 primary sex ratio. I actually think that the evidence is pointing towards the crappy habitats that are left after the good habitats are removed. You get this disproportionate number of females that are kicked into those habitats. Over time, their mortality is higher proportionally to males. And so I think that contributes to a higher mortality of females and a male bias sex ratio. I'm just guessing. I haven't done the math on that, but I'm guessing that might be one of the things that's happening.
Thank you.
I am so thrilled to hear about the Kirkland warbler. I used to work with them 12 years ago in Wisconsin.
Yeah.
And I'm so happy to know that they're doing better. So I'm
Now they
thrilled
are.
about all this work, but
Well, I got bad news for you. The Bahamas is going underwater. So we need to start worrying about the wintering grounds. Sorry,
okay.
not to
Well,
burst
I
your
mean,
bubble.
in this seasonal thing all makes sense. I mean, there's no doubt about it. You can't just think about one season because there's just too much in the life cycle going on all year round. So
Yeah.
thanks.
Thank you. Well, thank you very much. I'll be around all day and tonight. Thanks very much.